Hello students, today I am going to discuss about plant tissue culture and its applications. So just see the plant tissue culture lab. Now in plant tissue culture so many terminology are there. You know, tissue culture is the method of propagation of plant in the laboratory in artificial condition in artificial environment. We can regenerate the plant through micropropagation, through organogenesis, through somatic embryogenesis. So we can also say it is a method of micropropagation. Tissue culture technique is adapted to keep in pace with demand and qualities. Now the terminology in plant tissue that is de-differentiation. You know, the phenomena of reversion of the mature cell to meristematic state leading to formation of callus is called de-differentiation. Means you are going to convert the mature cell into meristematic cell. You know, meristematic cell, a cell that has ability to differentiate and ability to multiply. The component cell of callus have the ability to form a whole plant, and the phenomena described as redifferentiation means, you know, callus is undifferentiated growth of the plant cell in plant tissue culture media. Means we cannot differentiate whether it is a root part or a stem part, but we can multiply the cell in plant tissue culture media if you supply the auxin and cytokinin 1:1 ratio. Next, totipotency. It is the ability of a cell to regenerate whole plants. That is known as totipotency. Means from a small section of the plant leaf or stem or cotyledon, we can generate a whole plants. Such type of ability are generally we can observe in plant. Next, explant. You know, for the selection of the explant, we should consider whether it is not infected with any virus, bacteria, or fungus, and it should be new one cell. Generally, we take a meristematic root or stem. A small piece of the desired plant is selected. Generally, the meristematic tissue or internal segment of the plant is selected for micropropagation, and the selected plant tissue culture is called as explant for producing better hybrid, better uh, variety of the plants. Suppose this is explant, that is the meristematic portion. So you can select 1 or 2 centimeter like that for use as a explant during plant tissue culture media. Next, that is culture medium. Culture medium is the important factor that will decide whether that part will form the callus or that part will form the stem or that part will form the root. Okay, so this tissue is so collected is placed in a culture medium, nutrient medium for multiplication of the cell that contain all the organic supplements, vitamins, mineral, inorganic salt, and we can use as a agar as a solidifying agent. Okay, so that is the content of culture media. You can know agar gels, it act as a solidifying agents. Generally question asked why we use agar instead of gelatin, because agar will remain solid even 44 degrees celsius, but gelatin will not remain. And generally we use 1% agar for making plant tissue culture media, that is the solid media, but gelatin generally become liquid above the room temperatures. A carbon source is essential. Inorganic salt is essential for the proper growth and development of plants. Plant growth regulator mainly auxin, gibberellin, and cytokinin. You know, auxin responsible for root multiplication, gibberellin for internodal elongation, and cytokinin for the shoot developments. Just see the plant tissue culture plates. You can put the media, okay, and then we put the explant. Now callus, you know what is callus? It is undifferentiated growth of the explant in plant tissue culture media. So there are so many different type of media in which you put the small section of the plant, that is explant, to regenerate the who on large number of the cell, you cannot say it is the root cell or stem cell, but has ability to divide and multiply. Just see, the culture cell will grow into an organized mass that is known as callus. Callus grown in the nutrient media. The media start depleting because the that growing cell will absorb the nutrients, so many vitamins, so many minerals, so many organic source as a source of energy and inorganic salt that will provide the growth and development of the plant. So colors should be periodically changed into culture media for healthy growth of the plants. Okay, so callus is the important step in the plant tissue culture. Just see the different stage of the callus in the you can say in diagram A, B, C and D. Okay, so plantlets growing in the culture room in suitable environmental condition, temperature 25 plus minus 2, humidity 70 to 60 to 80%, and light duration minimum 16 hours, and it should be free from virus, bacteria and fungus. Only then plant will grow in healthy way. Next, the callus grow in artificial condition. You see the callus so grow up after differentiation is transferred into a small container for further differentiation for rooting and shooting. So nutrients and plant growth hormones are for the healthy growth. So plant tissue culture lab, that is the important part, where you put all the transferred explant or callus into a suitable culture room to make free from virus, and there will be the proper growth of the plant cell, either root part development or shoot part development in presence of suitable artificial environmental condition. Okay, so laboratory conditions should be completely aseptic, highly sterilized. That is the very important condition for plant tissue culture lab. Explant selectors should be sterilized before propagation. Before putting the explant in the media, you have to clean with the tap water. Then we can also use the mercury chloride for 1 second, or then again wash with distilled water. Then we put inside the jam bottles or culture plates for propagation. So these are the fully grown suma clone. You know suma clone, that is the each of these plantlet will be genetically identical to the each other. The plant parent plant from the which they are grown, they are suma clone, means they are genetically identical, because we are taking the plant cell as explant from the somatic cell, not from reproductive cell. Now these are the steps of plant tissue culture. You can observe, this is the plant in the pot. We have taken isolated cell, excise the piece of leaf as explant, put in the media, and then multiply in the bioreactor, and then you can change the media. 1 is to 2 ratio of oxygen and cytokinin, or 1 is to 1 ratio of oxygen and cytokinin, or 2 is to 1 ratio of oxygen and cytokinin. Decide the which type of plant cell will develop. Either callus will develop in 1 is to 1 condition of the ratio of oxygen and cytokinin, or stem will develop in 1 is to 2 ratio of oxygen and cytokinin condition, growth hormones, or rooting will start in media containing 2 is to 1 ratio of oxygen and cytokinin. You can get full grown plants, and then we transfer into the greenhouse gas, and then glasshouse gas, and then field condition. So just see, this is the diagram representing the step of plant tissue culture. Tissue sample scraped from the parents, then tissue sample placed in agar media as explant with nutrient having oxygen and cytokinin ratio in proper conditions. To make a callus, if you bypass the callus, then you have to change the media. Instead of 1 is to 1, you have to utilize 1 is to 2 ratio of oxygen and cytokinin for more proliferation of stem, and then further change the media for rooting or root proliferation. Plantlet will develop through this plant tissue culture methods. Just see the hardening. You know hardening means proper development of xylem and phloem for the absorption of nutrient by the new plantlets. So plants are transplanted into laboratory agri field for the better. Means it can survive in environmental condition. So we are going to acclimatize the plants gradually, slowly. First we put in the from the lab to greenhouse, then greenhouse to glass house, and then into the field condition. So adaptability before transferring into the main field, we have to pass the growing plantlet into a greenhouse, then further glass house, and then further other that is the field condition. Now, rapid multiplication of plant application. You know, rapid multiplication of plant through organogenesis, somatic embryogenesis, and micropropagation that are the method for regeneration of plants. You can also produce virus-free plant and also produce artificial seed. You know, artificial seed, it is encapsulation of calcium alginate or sodium alginates for rapid multiplication of plants. And you can also saving of the endangered plant species through embryo rescue. What is embryo abortion? Means chromosomal imbalance of the what that is endosperm that create a death of the plants. So we have to take the early stage embryo into plant tissue culture and multiply the plants. Then production of seedless variety of bigger size fruits, mainly in citrus and in apple or pear, and somatic hybrid and hybrid production we can also produce through hybridization and hybridization. You know, fusion between two nucleus and two cytoplasm, that is hybrid, and fusion between two cytoplasm and fusion between uh, one nucleus and other cytoplasm, that is hybrids. So production of secondary metabolite also a part for the production of for as a use as a medicine in through plant tissue cultures. So generally secondary metabolite it behave as a protective agent of the plants, but human beings are utilizing as a medicinal purpose. So production of edible vaccine and transgenic resistant variety plant against a biotic factor for improving the quality and quantity of crop production. Increase the shelf life of the fruits and vegetables. You know, flavor saver variety of tomato. You can know you can know the increase the shelf life and we can get the market value. That is the through antisense RNA technology and create the hybrid variety of crop through genetically main stability. Yes, so these are the examples. Artificial seed, you know the diagram. Outer coating, that is artificial seed coat. Inside you can get the somatic embryo and you know a somatic endosperm. These are the transgenic plants through different plant tissue culture methods. Then transgenic golden iron ice that contain iron and vitamin A contain gene and tearless variety of onions. And you can say the blue uh, blue rose, that is the creation of the and application of the plant tissue cultures. So thank you very much, and just utilize this content for your improving your content in the field of plant tissue culture, in the biology as well as in biotechnology. Thank you very much.